Good afternoon to all. So check once my audio and video are clear. All of you students, please check my voice, audio and video. Good evening, good evening. Good evening to all. Yeah. Okay, everything is clear. Right. Shall we start? Okay. In the last class, we discussed about the introduction to cellular respiration, right? Correct. What is cellular respiration? Why it occurs? Where it occurs? What are respiratory substrates? Is it all respiratory substrates yield same amount of energy? Is respiratory organs are required for respiration? In plants, no such respiratory organs, however they undergo respiration. Is microbes undergo respiration? Yes, microbes also undergo respiration. What happens during cellular respiration? The respiratory substances are oxidized with the liberation of energy. That energy is used to synthesize ATP. Oxidation of food material with the liberation of energy, and this energy is used to synthesize ATP, that is adenosine triphosphate, commonly called as energy currency. Correct? All these we discussed in the last class. Okay? And finally, we have seen regarding the differences between aerobic respiration and anaerobic respiration also. So respiration which takes place in the presence of oxygen is called aerobic respiration. Respiration which occurs in the absence of oxygen is called anaerobic respiration. In aerobic respiration, glucose is completely oxidized. As a result, carbon dioxide, water, and energy is produced. Whereas in case of anaerobic respiration, glucose, that is the respiratory substrate, is partially oxidized. Because of that, only less amount of energy is produced. I told you all these, right? Okay. Now today we will discuss about how the respiratory substrate is oxidized. I told you that it is oxidized in stepwise manner, not in a single step. In aerobic respiration, 4 steps are present. In aerobic respiration, four steps are present, namely 1. Glycolysis, 2. link reaction, three, Krebs cycle, and four, electron transport system. Electron transport system. Simply it is called ETS. Okay. So in aerobic respiration, four steps are involved during the oxidation of the respiratory substrate completely into carbon dioxide and water. I hope all of you are following well. Right? These are the four steps. Glycolysis, link reaction, Krebs cycle, ETS. Right? Similarly, in case of anaerobic respiration, only two steps are present. One, glycolysis, and two, fermentation. Okay. So, in anaerobic respiration, glycolysis and fermentation, only two steps are present. In aerobic respiration, four steps are present. Okay. So, we will discuss one by one. Now, let us see the first step, common step between aerobic and anaerobic respiration is this one, that is glycolysis. Look at this. Okay. It is the common step between the both. Okay. So today we will discuss about a detailed account of glycolysis. This is very important, not only NEET but also IPE as well. It is a set of biochemical reactions. It is a metabolic pathway. So let us see what happens during glycolysis, how it takes place, how the substrate break down, how many reactions are present, what are those reactions, and how many enzymes are involved, what are these enzymes, and ultimately what is formed at the end of glycolysis. All we will see slowly, one by one. You please concentrate one carefully, right? So glycolysis. The term glycolysis was derived from two Greek words, namely glykos means sugar, lysis means splitting of sugar molecule. The term glycolysis was derived from Greek words, namely glykos means sugar, lysis means splitting, or breakdown. Breakdown of sugar. Right. The scheme of glycolysis, the pathway of glycolysis was discovered by three German biochemists, namely Embeden, Meyerhoff, and Parnas. These three scientists worked out and discovered the scheme of glycolysis. They are German scientists. They are biochemists, namely Embeden, Meyerhoff, and Parnas. From the first letters of their names, see, first letters of their names, E from Embeden, M from Meyerhoff, P from Parnas are taken, it becomes EMP. Therefore, glycolysis is also known as EMP pathway. Glycolysis is also known as EMP pathway because Embeden, Meyerhoff, and Parnas discovered this pathway. For their honor, this process is also called as EMP pathway. Everybody should concentrate here. This is a very important topic. It is a metabolism. Right? So glycolysis is also known as EMP pathway. Correct? Yes. Why? Because Embeden, Meyerhoff, Parnas discovered the pathway, the scheme of glycolysis. Hence it is called EMP pathway. Okay, clear? Now, where does this glycolysis takes place? The site of glycolysis is cytoplasm. Cytoplasm. Where glycolysis takes place? I told you cellular respiration occurs in all living cells of all organisms. Therefore, in every cell, there is a cytoplasm. In every cell, there is a cytoplasm. So, glycolysis occurs in the cytoplasm. The site of glycolysis is cytoplasm. Sometimes you can also call it as cytosol. Nothing wrong. Cytoplasm or cytosol. Okay? Right. Next. In glycolysis, glucose is partially oxidized. Glucose is partially oxidized in glycolysis. This partial oxidation occurs in all the cells. All the cells. So glycolysis occurs in all living cells. It occurs in all living cells. Whether it is prokaryote, whether it is eukaryote, whether it is aerobic individual or anaerobic organism, whatever it may be, in all the organisms, in all the cells, glycolysis takes place. And moreover, it is the first step of respiration. It is the first step of respiration. It is the first step of aerobic cellular respiration. Okay. And moreover, it occurs in aerobic organisms and anaerobic organisms. That means it occurs in aerobic respiration and anaerobic respiration. Therefore, it is the common step. It is the common step of both aerobic and anaerobic respirations. All these are very important points. Okay. The site of glycolysis is cytoplasm. That means glycolysis occurs in the cytoplasm. No cell organelle requires. It occurs in all living cells. I told you, in all organisms. And it is the first step, whether it is aerobic respiration, whether it is anaerobic respiration, the first step will be the glycolysis. Next. And it is the common step. So glycolysis occurs in aerobic respiration and it also occurs in anaerobic respiration. Therefore, it is the first step and it is the common step. Whether it is aerobic or anaerobic, this always takes place in cytoplasm. And it is independent of oxygen. Very important point. It is independent of oxygen. What is the meaning of independent of oxygen? Whether oxygen present or absent. Whether oxygen is present or absent, glycolysis takes place. That means glycolysis does not depend upon oxygen. Are you following, everyone? Whether oxygen present or absent, glycolysis takes place. And glycolysis is independent of oxygen. Okay? Next. In glycolysis, the substrate is glucose or fructose. Glucose or fructose. The substrate is Glucose or fructose. This glucose or fructose undergo catalytic breakdown, catalytic process, and produces pyruvic acid. Pyruvic acid. One molecule of glucose break down into two molecules of pyruvic acid. Now, two questions are clear. Substrates of glycolysis is glucose commonly, and fructose also act as a respiratory substrate. And the end product of glycolysis is pyruvic acid. Glucose is a 6-carbon molecule which is split into two molecules of 3-carbon compound. So therefore, pyruvic acid is a 3-carbon compound. Okay. A 6-carbon molecule break down into 3-carbon molecules. Okay, clear? Next. This is a catalytic pathway. Breakdown pathway. 6-carbon molecule ultimately break down into two 3-carbon molecules. This entire process involves 10 steps or 10 reactions. 10 steps or 10 reactions. And it is a enzyme catalyzed process. Each step is catalyzed by one enzyme. Therefore, 10 different enzymes are involved. 10 different enzymes are involved. Okay. There are 10 steps in glycolysis process and there are 10 enzymes. Okay. Ultimately, through these 10 enzymes, stepwise manner, the glucose, the starting substrate, break down into ultimately the key product called pyruvic acid. End product of glycolysis, two molecules of pyruvic acid. Substrate is glucose. Okay, clear? I hope all of you are following well. If you want to become a doctor, certainly, indispensably, you have to follow, you have to learn, then only your dreams come true. Because it is a 2-year target. 2 years target. Success won't get in overnight. Every chapter you have to learn comprehensively. Then only your goals can be achieved. Okay? Please concentrate. Now let us see. So glucose break down into pyruvic acid. Let us see now. And one more point also there here. In plants, the end product of photosynthesis is sucrose. That means, I told you, glucose is the substrate for glycolysis. In plants, that glucose comes from the end product of photosynthesis called sucrose. Sucrose is a disaccharide. Sucrose is a disaccharide. Okay. Or storage carbohydrate. Storage carbohydrate, that is starch. That means the glucose in plants comes either from sucrose or from starch. Starch is a polysaccharide. Sucrose is a disaccharide. Okay. So in plants, the glucose or fructose comes from these two end products of photosynthesis. So the direct product of photosynthesis is sucrose, and this sucrose is converted during storage in the form of starch. Storage product. Now let us see. This sucrose break down into two monosaccharides, namely glucose and fructose. Glucose and fructose by an enzyme called invertase. By an enzyme called invertase. Okay. Sucrose, end product of photosynthesis in plants, which is a disaccharide. This disaccharide split into two monosaccharides. Remember, these two are monosaccharides. Okay. Glucose and fructose are monosaccharides. Now, this glucose or fructose act as substrate for glycolysis, more commonly the glucose. Similarly, glucose is also formed from the starch. Okay, that's all. Now let us see how this glucose break down. As I said, the substrate is glucose. Okay. Glucose, a 6-carbon monosaccharide. First, converted into glucose 6-phosphate. Glucose 6-phosphate. This step is called phosphorylation. That means ATP is used so that it become ADP. That means one phosphate in the ATP is used during this conversion so that ATP become ADP, and that phosphate is added to glucose so that glucose becomes glucose 6-phosphate, because at 6 carbon that phosphate is added. Glucose 6-phosphate. This is the first step. Correct? Glucose, starting substance, the substrate undergoes phosphorylation and form glucose 6-phosphate. This reaction is catalyzed by an enzyme called hexokinase. This is catalyzed by an enzyme called hexokinase. That is the first reaction. Next. Okay, first step is over. Glucose is phosphorylated to glucose 6-phosphate. Here, one ATP is used. One ATP is used so that it become ADP. Clear? And this is catalyzed by an enzyme called hexokinase. Yes. Next. In the second step, this glucose is isomerized into fructose 6-phosphate. Fructose 6-phosphate. This is 6-carbon molecule. This is also 6-carbon molecule. This is also 6-carbon molecule. Now glucose 6-phosphate is isomerized into fructose 6-phosphate. This reaction is called isomerization reaction. And this is catalyzed by an enzyme called phosphohexose isomerase. Simply I am writing as isomerase. Hope you understand the second reaction also. Second reaction name is isomerization. Glucose 6-phosphate is isomerized into fructose 6-phosphate. May I slow down? No. Okay, I will continue like this. Right. So in the first step, glucose is phosphorylated to glucose 6-phosphate. In the second step, glucose 6-phosphate is isomerized into fructose 6-phosphate. I told you in the beginning, either glucose acts as substrate or fructose acts as substrate. If glucose is act as substrate, it will continue like this. If fructose acts as a substrate, that fructose directly phosphorylated to fructose 6-phosphate. Got the logic? Do you understand? Do you understand that logic? If glucose is the substrate, two steps are required here. If fructose is the substrate, fructose is phosphorylated directly into fructose 6-phosphate. The remaining steps are same. If fructose is taken, 9 steps only comes. If glucose is taken, 10 steps comes. Understand? Because of the difference here only. Only here the difference. Okay, right. This step is phosphorylation. This step is isomerization. This step is catalyzed by hexokinase. This step is catalyzed by isomerase. In this first step, ATP is used. Okay. Now, next step, third step. Fructose 6-phosphate is again phosphorylated to fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. Here, once again, ATP is used. ATP is used so that ATP becomes ADP. This is catalyzed by an enzyme called phosphofructokinase. Phosphofructokinase. This is the third step. What is the third step? Again phosphorylation. Second phosphorylation. Addition of ATP is called phosphorylation. So already one phosphate is there at the sixth carbon. One more phosphate is added from ATP here at the first carbon. So that now two phosphates are there on the fructose 1,6-bisphosphate. So this second phosphorylation product is fructose 1,6-bisphosphate. This reaction is also phosphorylation. In the sequence you can say second phosphorylation, because this is the first phosphorylation where one ATP is used. It is the second phosphorylation. Here one more ATP is used, and this is catalyzed by a pacemaker enzyme called phosphofructokinase. Phosphofructokinase. Yes, I told you the shortcut name. Phosphohexose isomerase. Simply I am writing isomerase, nothing wrong. Okay, absolutely right. Next, how many steps completed so far? How many steps completed so far? 3 steps completed. Next, fourth step. This fructose 1,6-bisphosphate. Very good. 4 steps. 3 steps, sorry. 3 steps. Now we are going to start the fourth step. Fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate split into two molecules of 3 carbon compounds. 3 carbon molecules. One is 3-phospho glyceraldehyde. 3-phospho glyceraldehyde. In shortcut, you can call it as PGAL. PGAL means phosphoglyceraldehyde, and dihydroxy acetone phosphate. Dihydroxy acetone phosphate. Dihydroxyacetone phosphate. In shortcut, you can call it as DHAP. DHAP. Dihydroxyacetone phosphate. Shortcut name. So PGAL 1 molecule and DHAP 1 molecule are formed. So this is 6 carbon. Remember, all are 6 carbon molecules now up to this. All are 6 carbon molecule. Now, one 6 carbon molecule split into 2 3 carbon molecules. This is 3 carbon. This is also 3 carbon. Okay. One 6 carbon molecule split into 2 3 carbon molecules, namely PGAL and DHAP. These 2 are isomers. These 2 are isomers. You can also call these as triose phosphates. This is one triose phosphate, this is another triose phosphate. Both are triose phosphates. Okay, 3 carbon compounds. These two are isomers. These two are catalyzed by triose phosphate isomerase. And you remember here, the splitting of fructose 1,6 bisphosphate into 2 triose phosphate is catalyzed by aldolase enzyme. I am writing here. This is aldolase. And here also I will write it a different color so that you can identify. Isomerase. Yes. This is another very, very important reaction. This reaction name is called cleavage or splitting. A 6 carbon molecule split into 2 3 carbon molecules. One PGAL, one DHEAP. These two are isomers. Again, they are interconvertible. These two are isomers. This isomerization reaction is catalyzed by triose phosphate isomerase. In shortcut, I wrote as isomerase. This splitting reaction is catalyzed by aldolase. Next onwards, you should be very meticulous because 2 compounds are produced here. 2 molecules are obtained. So you have to count 2 in every step because DHEAP converted into PGAL and then continue the further reaction. That means from here onwards 2 molecules are involved. Am I right? Yes. Now, what happens here? Here, one oxidation reaction takes place. Next reaction is oxidation. I will recollect once again. Try to recollect all the steps. Phosphorylation, isomerization, again phosphorylation, next splitting or cleavage, next isomerization, and next step is oxidation. Next one is oxidation. Let us see what happens here. The PGAL undergoes oxidation followed by phosphorylation from inorganic phosphate and form 1,3 bisphosphoglyceric acid. In shortcut, you can call it as BPGA. 1,3 bisphosphoglyceric acid. Here NAD plus becomes NADH. See, 2 NAD plus becomes 2 NADH. These are coenzymes. Coenzyme NAD plus is reduced into NADH. Why you have drawn 2 here? Because this one molecule, 2 molecules are involved in the further reactions. That is why. Here also 2 molecules. Okay. And here phosphorylation also occurs. See, 3-phosphoglyceraldehyde. Only 1 phosphate is there. Here 1,3-bisphosphoglyceraldehyde. That means 1 phosphate is given. Who is the donor of that phosphate? Is H2PO4. Inorganic phosphate is involved here. That means H2PO4, inorganic acid, is the donor of phosphate in this reaction. Okay? And this reaction is catalyzed by an enzyme called G3P dehydrogenase. Simply I am writing as dehydrogenase. Dehydrogenase. Okay. This is complex step. This is the oxidation reaction. So end product of oxidation is NADH. How many NADH are formed? 2 NADH are formed. Because for one PGAL, 1 NADH, 1 DHAP, after converting into PGAL, 1 more. Therefore, totally 2 NADH are formed. So the product of oxidation reaction is the NADH. Okay, here also phosphorylation occur, but ATP is not the donor. The donor of ATP phosphate is the inorganic phosphate. Inorganic phosphate. Definitely, this is somewhat a biochemistry. So therefore, you have to concentrate carefully. Then only you will understand. That is why I am telling repeatedly. The terms, the terminology, and all these are using number of times. Okay, right. So, after the 6th reaction, 1,3-bisphosphoglyceric acid is formed. Right? Next, what happens? 1,3-bisphosphoglyceric acid undergo dephosphorylation and form 2 molecules of 3-phosphoglyceric acid. 3-phosphoglyceric acid. Simply you can say 3-PGA. 3-PGA. Here, reverse to these 2 reactions. ATP is formed. ATP is not used. ATP is formed in this step. How many? 2. 2 ADP becomes 2 ATP. 2 ADP becomes 2 ATP. Who is the donor of those phosphates? 1,3-bisphosphoglyceric acid. So previously it has 1 phosphate on first carbon and another phosphate on the 3rd carbon. Now it become only 3-PGA. That means 1 phosphate is added to ADP, so that ADP becomes ATP. So here ATP consumed. Here ATP is formed. During conversion of one substrate into another substrate, if ATP is formed, this reaction is called substrate level phosphorylation. This reaction is called substrate level phosphorylation. SLP reaction, you can call it as in a shortcut. SLP, substrate level phosphorylation, SLP reaction. So in SLP reaction, ATP is directly synthesized, not in the last step that is ETS. So if ATP is directly synthesized during conversion of one substrate to another substrate, it is called as SLP reaction. Okay, this is catalyzed by an enzyme called phosphoglycerokinase. Phosphoglycerokinase. Phosphoglycerate kinase. Okay. Next, 3-PGA becomes 2-PGA, 2-PGA, 2-phosphoglyceric acid, 2-phosphoglyceric acid, 2-PGA in shortcut. So intramolecular shift, phosphate is present at the third carbon, right? Now that is shifted from third carbon to second carbon. This is called intramolecular shift. Because of that, the positions of phosphates are changed. This reaction is catalyzed by an enzyme called phosphoglyceromutase. I am writing the shortcut name mutase. Phosphoglyceromutase. Okay. I think you may be confused here. This is the area where you have to focus. I repeat again, don't worry. I repeat. After completion of 2 more steps are there. Now, 2-PGA converted into 2 molecules of 2-PGA is converted into like this, 2 molecules of phosphoenol pyruvate or pyruvic acid, simply called PEP. Phosphoenol pyruvic acid. 2 molecules of phosphoenol pyruvic acid. Here dehydration takes place. Phosphoenol pyruvate is formed. This is catalyzed by an enzyme called enolase. Name of the enzyme? Enolase. Okay. And finally, last step is again dephosphorylation reaction. Last step, that is dephosphorylation. Here becomes 2 molecules of pyruvic acid, a 3-carbon molecule. This is catalyzed by here also very important, 2 ATP are formed. Here also 2 ADP becomes 2 ATP. Very, very important. In the first step, 1 ATP used. In the third step, 1 ATP used. But here in the 7th step, 2 ATP formed. In the 10th step, 2 ATP formed. So, here 1 and 3 reactions, 2 ATP used. In 7th and 10th reactions, totally 4 ATP formed. And finally, pyruvic acid is formed. And this reaction is catalyzed by an enzyme called phospho or pyruvate kinase. Simply you can write enzyme. And this is the end product of glycolysis. That is the end product of glycolysis. So, substrate for glycolysis glucose or fructose, and the end product of glycolysis is pyruvic acid. Okay. So, how many steps are involved in glycolysis? Totally 10 steps. Okay. 10 steps. I repeat all the steps once again fastly. Please focus on this. First, see, glucose is phosphorylated into glucose 6-phosphate. Here ATP used so that it become ADP. Next, this is catalyzed by hexokinase. Second reaction, this glucose 6-phosphate is isomerized into fructose 6-phosphate. This is the second step, isomerization reaction. The enzyme involved is phosphohexose isomerase. Simply I wrote as isomerase. Next, fructose 6-phosphate is phosphorylated again into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Here also 1 ATP is used. The enzyme involved is phosphofructokinase. Okay. Now, this fructose 1,6-bisphosphate splits into one molecule of PGAL and one molecule of DHEAP. One molecule of fructose 1,6-bisphosphate split into one molecule of PGAL, one molecule of DHEAP. PGAL means phosphoglyceraldehyde, DHEAP means dihydroxyacetone phosphate. These two are isomers. Okay, the splitting reaction is catalyzed by aldolase, and these two are isomers. Those are converted by an enzyme called isomerase. And now here you have to focus, right? PGAL undergoes oxidation and form followed by phosphorylation, but the for phosphorylation donor is not the ATP but is the inorganic phosphoric acid, that is H2PO4, right? This PGAL becomes 1,3-bis-PGA, simply called BPGA. This is an oxidation step. The product of oxidation is NADH. 2 NADH are formed in glycolysis in this reaction. This is catalyzed by an enzyme called dehydrogenase. Next, 1,3-bis-PGA, 2 molecules. From here onwards, you have to consider 2 molecules because here 1 molecule and 1 molecule. 2 are simultaneously involved. Right. 1,3-bisphosphoglyceric acid undergo dephosphorylation and 3-PGA are formed. Here ATP are formed. In this case ATP are used. Here ATP are formed. Totally 2 ATP are formed. For 2 molecules 2 ATP. This is catalyzed by phosphoglycerate kinase enzyme. Next, see, this 3-PGA becomes 2-PGA. 3-PGA become 2-PGA. This is an intramolecular shift. The position of phosphate from 3rd carbon changed into 2nd carbon. That is why it become 3-PGA become 2-PGA. Now this 2-PGA, this is catalyzed by mutylase, and 2-PGA become 2-PEP. 2-PGA become 2-PEP. PEP means phosphoenol pyruvate. Here dehydration occur. Water molecule is removed. Dehydration occur, that is water molecule is removed. And finally, the PEP, 2 molecules of PEP, again undergo dephosphorylation and become 2 molecules of pyruvic acid, the key product of glycolysis. The key product of glycolysis is pyruvic acid. The key product of glycolysis is pyruvic acid, and pyruvic acid is the end product of glycolysis, and it is a 3-carbon molecule. And during this process also, 2 ATP molecules are formed. 2 ATP molecules are formed. Okay? And this reaction is catalyzed by an enzyme called pyruvate kinase. Look at this. Whenever kinase name will comes, where ATP either formed or used. If the enzyme name has kinase, right? So here 2 ATP, here 2 ATP formed, 4 ATP. Here 1 ATP, here 1 ATP used. So net gain of ATP will be 2. Net gain of ATP will be 2. Okay? See, in glycolysis, no oxygen consumed. Okay? Oxygen is not consumed. No CO2 liberated. Yes? So it is independent of oxygen, I told you. Whether oxygen present or not, glycolysis takes place. Right. Next. Number of ATP used, 2. Number of ATP formed, 4. Number of NADH 2 formed, 2. These are very, very important. Therefore, net gain of ATP is equal to 4 minus 2, that is equal to 2. This is glycolysis in a nutshell. Oxygen is not consumed, CO2 is not liberated, number of ATP used are 2, number of ATP formed are 4, so net gain of ATP will be 2 and number of NADH formed are 2. So this is about glycolysis, students, and we will continue what is the what is the fate of pyruvic acid. I told you pyruvic acid is the key product of glycolysis. The fate of pyruvic acid depends upon cellular need. What happens? How this pyruvic acid enters into aerobic or anaerobic respiration. All these we will see in the tomorrow's class. I hope you understand, everyone. Okay? That's all today. Thank you. And continue in the next class.